Hello, everybody. In this lecture, we're going to talk about the extracellular structures of eukaryotes. So let's go ahead and get right into it. We got a bunch of objectives here, but they're all fairly straightforward. By the end of this module, you should be able to discuss the basic structural and functional features of the eukaryotic plasma membrane, their cell walls, the glycocalyx, um, their flagella, compare their flagella to prokaryotes, and also the structure and function of cilia. And so, when we think about the eukaryotic plasma membrane, they're very similar to prokaryotic plasma membranes. All cells, in fact, have a plasma membrane. It's pretty much the defining characteristic of, or one of the defining characteristics of a cell. Just like in prokaryotes, they're a selective barrier. They participate in, uh, passive transport, active transport, all that good stuff. It's a phospholipid bilayer, just like in your prokaryotes, using either your saturated or unsaturated fatty acids. One of the things that's different is that there are sterols in your eukaryotic membranes, and particularly in animal cells, we have cholesterol. Um, Things like cholesterol and other sterols are good for maintaining membrane fluidity. They keep it more fluid when it's cold. They keep it from getting too fluid when it's warm. So they kind of function both directions. Some eukaryotes have cell walls, but not all of them do. These are external to the plasma membrane, so they're not like they're more akin to a gram-positive cell than they are to a gram-negative, in that there is a single membrane, and on the outside of that membrane is your cell wall. And like prokaryotes, the eukaryotic cell wall is going to help maintain cell shape, protect against mechanical and osmotic stress, and shear forces. However, unlike uh, prokaryotes, they do not have peptidoglycan. Their cell walls of the different types of eukaryotes are made of all sorts of things. And fungi, they're made of chitin. And in plants, it's made of cellulose, which is the most common biopolymer on the planet. In your protists, it can be all over the place because protists aren't really a good kingdom, because they should be probably like 4 or 5 kingdoms. But in these algal cells, we see that they have cellulose similar to plants. But in these diatoms, we have like this completely different type of cell wall, which is made of silica, which is an inorganic molecule, and it ends up making them very pretty. Eukaryotes have other external structures which can be used for cellular protection, adhesion, and motility. When we're thinking of the glycocalyx, this is very similar to the glycocalyx of a prokaryote. So this is going to be used for adhesion. It's going to be made up of primarily carbohydrates, glycoproteins, and glycolipids. It allows to adhere to other cells or to surfaces. When you adhere to other cells, you might be able to communicate with them. Now, it's a glycocalyx, so keep in mind the glyco prefix that means that essentially means sugar or carbohydrate, and a glycoprotein is a protein with a carbohydrate attached, and a glycolipid is a protein or is a lipid with a glyco or with a carbohydrate attached. So the flagella of prokaryotes and eukaryotes are quite different. In prokaryotes, the flagella is made from the flagellin protein. However, in eukaryotes, there's a tubulin protein. In the prokaryotes, they don't have microtubules, but they do in eukaryotes, and they have this 9+2 arrangement, which we'll talk about in a moment. In prokaryotes, the generally speaking, the flagella is not membrane bound. And what that means is that it actually is inserted through the membrane in the cell wall. It is not surrounded by membrane. However, in eukaryotes, it is. And in the anchors, that hook and filament we talked about when we talked about prokaryotes, but for eukaryotes, They actually sprout from a basal body, which is a centriole. You might know centrioles from like mitosis and stuff like that. Um, and the motion is different as well. So the motion of a prokaryotic flagella is rotary, so it kind of like spins around like a propeller. However, in eukaryotes, it's wave-like, so it whips back and forth. And so here is a picture of that 9x2 microtubule setup, 9 on the outside, 2 on the inside. It's anchored to this centriole basal body inside the cell. And when it moves, it's going to flip side to side. And the classic example for this is the sperm cell. If you've ever seen a video of a sperm cell swimming, it's just kind of like whipping that tail side to side in this wave-like motion. Um, Unlike your bacteria, which are going to have a spinning or rotary motion. Cilia also are involved in movement. There are many cilia on your eukaryotic cells, and they are kind of short. They look kind of like hairs, and they actually are going to move in an oar-like fashion. So if you think about how when you're rowing a boat, or if you've ever seen someone row a boat, you've got the power stroke, which is the initial kind of pull backwards, and then when you have the recovery stroke, which is where you pull the oar out of the water, come forward, and then put it back into place. And so they kind of move in concert using that power stroke, recovery stroke back and forth. And so with that, you should now be able to discuss the basic structure and functional features of plasma membranes, cell walls, the glycocalyx, the flagella, and cilia, as well as comparing that some of those functions with how they work in prokaryotes as well as eukaryotes. I hope you enjoyed, and I'll see you soon.